Can aging process be slowed down? Have you ever wondered why some people look younger at their age while some look older at their age? Hi, my name is Delbert. I'm a student intern at Snow Hill Science in Cyberport, Hong Kong. Today I'd like to share an elusive yet important concept of biological age. When we talk about age, we usually refer to the chronological age. Then what is biological age? How much do we know about this concept? And by knowing this, how can we potentially utilize it as a measure of individuals' functional capacity? And finally, with this concept in mind, is there a way we can address aging on one hand and promote longevity on the other hand? Everyone's physical and mental functioning gradually declines from early adulthood onwards, but this happens at different rates in different people. This therefore makes chronological age an inaccurate measure of the functional capacity of an individual. How do we measure biological age? We owe much of our understanding of this concept and beyond through the seminal work of Steve Horvath on epigenetic clock. Epigenetic clock is based on the methylome, or the genome-wide DNA methylation that appears to be concentrated at certain regions enriched with guanine and cytosine nucleotides, which we often refer to as the CpG islands. Methylation is an enzymatic and reversible process that adds methyl groups to CpGs, which thereby affect gene expression dynamics, offering a clear mechanism by which it can affect biological function. DNA methylation is most commonly associated with downregulation of gene expression. However, positive associations of DNA methylation to gene expression have also been reported, suggesting a more diverse mechanism of epigenetic regulation. In other words, methylation can turn on or turn off gene expression. The positive and negative impact of epigenetic changes can be summed up as collective temporal changes that affect gene regulation. This idea of shifting DNA methylation profiles reinforced the idea of epigenetic signal as a function of aging. But how such epigenetic signals become useful in the biomedical perspective? It becomes useful when such signals are associated or linked with certain phenotype, predicting some functional outcome. Here, the occurrence of age-related illnesses and lifespan. So we are essentially looking at deviations from chronological age. Interestingly, only a small fraction of the total CpG islands in the human genome provide useful signals. Unlike chronological age, which has a constant rate of change, biological age is believed to be driven by an evolutionary conserved developmental process across mammalian species. Meanwhile, it has also been shown that aging results to a gradual loss of cell identity induced by repeated cycles of stochastic DNA lesions and repair mechanisms, which result in genome-wide DNA methylation changes. Finally, if we can somehow calculate the deviation of biological age through the epigenetic clock with respect to an individual's chronological age, then perhaps interventions can be set in place to address accelerated aging while promoting longevity.